that is going to be assisting us in our very first webinar to educate the community about what regenerative medicine can do for cerebral palsy and how it technically works. And we're going to probably have about 30 to 40 minutes of a presentation, and the floor will be left open for about 20 minutes for question and answer. And I'll let Kevin Metzger, who's running this webinar, take over now so that he can go over the outline of the webinar.
Okay. So just the way we're running this, we will be muting everybody. I don't have everybody quite muted yet, but we will shortly. And if you have questions during the presentation, there's a chat box at the top of your screen that you can click on that says chat, and it should pop up a little box, and you can enter in questions through that chat box. You should maximize your screen so that you can see the presentation with the best resolution. I think there's something in the lower right-hand corner of your screen that will allow you to maximize the web export.
Actually, if you click on the right side of the screen, if you right-click on it, it will say View, and then you just go over to the View and click down to Fit to Width, and that will open it up a little bit wider, and the screen will be much larger.
Okay. And yeah, my screen's set up a little bit differently than the participants, so I couldn't see that, so thanks, Ed. Where we can, and there's a lot of definitions in here, we'll throw some into the chat box. I've gotten as many as I can up to this point, but I'm not sure if we have all of them, but we'll try and make sure that we define it as best as possible as we're talking to it, and Cliff will be sure about that. If you have questions, you should be able to raise your hand when we open it up for questions, and we'll go ahead and pull you off mute and try and allow for questions that way. I kind of went over what the agenda is going to be already, so I'm going to actually go ahead and put everybody fully on mute. Are there any questions right now? If there aren't, everybody's off of mute for right now. So if you have any questions, go ahead and speak now. Put everybody on mute and just unmute Ed and Cliff and Lizette, and they'll get us underway. So just give us a second, Ed and Cliff and Lizette, and then I'll hand it over to you. Okay, Ed, are you there?
Yes.
Lizette?
Yes, I'm unmuted now.
All right, Cliff?
I'm here. My client's saying it's connecting me to the meeting, but it's just sitting there.
So you don't have the screen up yet?
Not yet.
Ed, do you want to go ahead and introduce your CP?
Sure. First of all, I'd like to thank everybody for joining our first webinar with Let's Cure CP. And I first want to give everybody a little bit of background about Let's Cure CP. Between the Denays and us, we co-founded Let's Cure CP a little over 1 year ago in 2010. And our board member consists of parents of children with cerebral palsy. And we are committed to finding a cure to improve our children's abilities. Myself, I've been in the medical industry, and I've run my own medical company for over 8 years. And Lizette has an extensive knowledge of the stem cell industry. She currently works in it, and her father is actually head of neonatology at a well-known institution in the United States. Right now, we are currently funding 2 regenerative medicine studies in the United States. We are currently funding Dr. James Carroll at Medical College College of Georgia, and Dr. Baumgartner we're going to be funding as well. We are committed to supporting these studies, and we want to make sure that they're done safely and that they will advance medicine for cerebral palsy in general. And currently, Let's Cure CP has no employees, so all the available funds that we have go directly to support our researchers. Next slide, please, Kevin. So with the support of the community, what we would like is if you can tell your friends, your therapists, and your doctors about what we're doing. This webinar that we're presenting right now is going to be recorded and be put onto our webpage so that you can direct other people to view this to get more educated about what regenerative medicine can do for our children. You also can donate directly to letscurecp.org or on our Facebook page, or if there are people out there that would like to mail checks, our P.O. Box is 81993 Atlanta, Georgia, 30366. A lot of people can, if they work for large companies, oftentimes, if you ask your company to support nonprofits, they're more than happy to do that. And then there have been people that have just gone out and done their own fundraisers in their own town to help support us, which goes directly to our researchers, all the way down to very small things in your local community that you can do to help. And we have people at Let's Cure CP that will help you get started if you want to help support us in any way. If you do, please just on our webpage, contact us, and we're more than happy to respond quickly. Lizette, do you have anything to add to that?
No, I think that's it for now. And, as Ed just stated, we are completely volunteer-driven at this point. So that's another nice thing. All the funds really do directly go to these important research studies that we're going to talk about tonight.
Cliff, is your computer ready to go?
No. Let me try the client.
Work off of your presentation that you have, and Kevin will go through to match it.
Let me try one more thing.
Can you hear us, guys?
I'm sorry?
Can you all hear us? This is Fran in Tampa, but I have to listen to it on the cell phone. It's not coming in on the laptop.
Okay.
Oh. Yes, and we can hear you.
Yeah.
Who's this, Lizette?
Yes.
Fran,
Hi.
Fran.
Lizette and Kevin. Hi, everybody.
How are you? Fran, I'm going to actually read you so that you can listen in, but we're trying to actually, for presentation purposes, just keep everybody on mute. As people come in, though, I have to mute them. Unfortunately, I don't have a setting right now to have everybody mute as they come in. So.
Well, I won't interrupt y'all. It's just I can't get it on the laptop. That web, whatever, didn't work.
Oh, okay.
Let's see.
Cliff, whenever you're ready, let me know, and what I can do while we're waiting for your presentation to load, I'll tell you a little bit of background about more background about Let's Cure CP. If you've been to our website, we have a philosophy page that it really explains what our organization is all about. We want to try to fund something that's going to be a solution to cerebral palsy to give our children greater abilities. And Kevin just pulled that up for us, so you can see that right there. There's some words there that people may not understand. One is allogenic, and there's autologous and cord blood. Most people are familiar with cord blood, which are the cells that are stored from the umbilical cord. And Dr. Kurtzberg and Dr. Carroll are actually both performing cord blood cells in the United States on cerebral palsy patients. Right now, I think the national average is about 3% of cord blood is banked. and so that leaves 97% of the CP population at best that doesn't have that ability. So Lizette and I wanted to expand that, and we talked to Dr. Carroll, and Dr. Carroll is actually working with allogenic cells, which is on the far left of your screen. And what an allogenic cell is they're adult bone marrow cells that are harvested from healthy adult individuals' bone marrow out of their hip. And there are companies that take these cells and they treat them so that they are the recipient's body will treat them as like a type O blood, where it won't be rejected and will go in very efficiently and cleanly without having to suppress the body's immunization so that anybody can receive these cells. There are other avenues in medicine that are using that. The cardiology field is using it for patients with stroke, and after heart attacks, they're delivering these cells, and they're stored in just little vials. So allogenic cells are extremely important for what we're doing with cerebral palsy, because there's a large percentage of cerebral palsy, well, there's probably about a 20% population of cerebral palsy patients that have what's called a degenerative disorder as well. So that means the own cells that that particular patient has will not be sufficient to heal their own body, and that's where the allogenic cells are so important. The autologous cells, which are in the middle, those are cells that are harvested from your own body. So what Dr. Baumgartner is going to be doing is that he will harvest these cells out of your own the cerebral palsy patient's own hip bone, and then process the stem cells and just re-administer those cells directly back into the patient through a simple IV. So the patient's getting his own stem cells. It's very clean, efficient, and it doesn't require any immunosuppressants, and the patient is getting their own cells. So between these three different types of cell lines, there is a great amount of hope for our own children, and right now there's probably been over 300 patients already treated with cord blood at Duke with cerebral palsy, and they have certain patients that are doing extremely well. There's some patients that are doing better, and some that they're trying to figure out if there has been much improvement. But the thing is, we're learning from this, we're growing from it, and there are patients that are getting extremely better from these cell treatments. And so with these physicians studying this type of medicine, our hope is that one day that every child with cerebral palsy will be able to be treated to make themselves better in the future. Cliff, are you ready to go?
It looks like the problem is the password. I have the wrong password, apparently.
Yes, I think, Cliff, you might have had the password I had initially. Let me give you the correct password, because what happened was we did get a wrong one.
The passcode is CP, lowercase, but then there is also a number that was to get into the phone call. Kevin, do you know what he needs to do?
It's CP. Just put in meeting password, CP in lowercase letters. Cliff, do you have the correct meeting number?
Yes, we already corrected that. That's how I managed to call in. That's why I was suspicious of the password. Let's see what happens here.
Yep.
Does anybody have any questions so far?
I just joined in the meeting a few minutes ago, and I'm not really sure. I'm not really up to speed on what you have already covered, but I may not be able to stay on the whole time. But what exactly have you guys covered already?
Well, we just basically introduced Let's Cure CP as an organization, and cover the real quickly, our philosophy page. We're waiting for our presenter Cliff's presentation to get loaded onto his computer right now.
Okay.
We're having a little bit of technical problems.
And guys, apologize.
Allogenic cells, autologous cells, and cord blood cells that can be used to help improve our children with cerebral palsy.
Okay.
If anybody else has a question, if you can raise your hand, I can unmute you. Let's see, just one other thing I'd like to, if you're not already a fan of Let's Cure CP, I'd like to just encourage you to go out to facebook.com/letscurecp and come and join in. We're trying to do a few things to try and involve the audience and involve all the members. One of the things we just started this past Friday is kind of the idea of a Friday picture theme that we're kind of start running, and once a month we'll pick one of the pictures that were posted to the page that week and actually bring it up into this area to make it part of the profile pic. And we're just kind of doing that to get to know everybody and get everybody involved in the community. So we're looking forward to having folks participate in that sense.
So, Cliff, if you can't get your into the meeting, go on from your presentation, and your slides should match the slides that Kevin has.
Yeah, I'm in now. So I don't know how. Yeah, there we go.
Okay.
My presentation's up now. Will I be advancing the slides?
I'll go ahead and do it, Cliff. Just we'll start there.
All right, sounds good. Well, again, my name is Cliff Wimborne, Harold Wimborne. I work with Dr. James Carroll at the Medical College of Georgia, which has just been renamed Georgia Health Sciences University. So if you hear either name, they are the same institution. And we just wanted to lay the groundwork here as far as what is cerebral palsy, how does it work, and what are we doing, what can we do as far as treating it, what are we looking into as far as research approaches. So let's go ahead and flip to the next slide. First, let's look at cerebral palsy, the actual definition of it. There's actually a group of disabilities associated with cerebral palsy. They're all motor-related, and they're non-progressive, which means that the underlying problem doesn't get worse. However, as was mentioned earlier, a lot of patients with cerebral palsy will also have other disorders as well. But the cerebral palsy itself, the brain damage that is present, does not get worse. Sometimes symptoms will show up later. You have developmental problems, but the core problem doesn't get worse. The other side of that, of course, is that it does not get better without treatment. And right now we're seeing about 2 to 2.5 cases per 1,000 live births. That's actually increased by about 20% in the last decade or so. Let's flip to the next slide. Cerebral palsy, the same symptoms can occur from different causes. 70% of those causes are prenatal, which means they occur during development, before labor and delivery. 20% are due to perinatal causes. That's injuries like preemies will be born with inadequate lung function, and so they will be hypoxic. They won't get enough oxygen to the brain. Also, you can have the umbilical cord could be wrapped around the baby's neck, and that can cause birth hypoxia. And there are other causes as well. And also in newborns, in about 10% of cases, the injury is in a newborn. That can be a physical trauma or some other event. Next slide, please. In addition to the muscle control problems, we often see speech problems, seizure disorders. About half of patients have some form of mental retardation. A third have some degree of deafness, and visual impairments are also common. And the reasons for all of these we'll see when we cover neuroanatomy a little bit later in the presentation. Let's go to the next slide. And as I mentioned before, as CP patients age and develop, those underlying impairments give rise to developmental delay. Not being able to move as well as would be ideal can limit a child's experiences, and that can lead to developmental delay. Also, improper muscle tone in severe cases can lead to skeletal malformations, which may be painful. Next slide, please. There are three primary types of CP that we'll see. The vast majority of them are spastic, which is due to upper motor neuron damage. Now, those are the neurons that start in the brain. There's actually a series of neurons that travel from the brain out to each muscle. But the upper motor neuron damage is actually in the brain. And what you get is the muscles are very, very tight. They can't be moved. There are cases even when the physician doing an examination will have trouble extending the limb because the muscles are so tight. And that can be extremely painful. The other more rare cases, you see ataxia, which is uncorded muscle control. That's due to damage in the cerebellum, which is that large, there's two lobes to the structure just below what most people think of as the brain, right at the back. And the cerebellum is really for fine-tuning muscle control. Damage in the cerebellum prevents, it causes certain speech disabilities and fine muscle control. And the final type is the dyskinetic, which gives you differing muscle tone in different areas of the body. And that's very difficult to treat, but thankfully it's a fairly rare form of CP. Let's go to the next slide. Okay, there are two types of brain damage primarily in CP. There's selective neuronal necrosis. Now, necrosis is cell death, but it's in an uncontrolled manner. The other type of cell death is called apoptosis, and that is a chemical process for signaling which basically shuts the cell down and marks it for the body to remove it. But with necrosis, the cell just dies, and there's no warning system for the body to clear that up. And so the cell contents can leak out. It can cause inflammation and other problems because all of those cell contents have now leached out, and you've got improper signals in that general area. The other type of brain damage we see in CP is PVL, paraventricular leukomalacia, which is more common in premature infants. That type of damage, it basically looks like cysts next to the ventricles, which are the spaces in the brain that hold the cerebrospinal fluid and assist with circulation. And I'll show you on the neuroanatomy slide why that's a particular problem. But PVL is essentially named for soft brain because you get these large holes, fluid-filled cysts next to the ventricles. Let's go to the next slide. Now we're going to go through a little bit of neuroanatomy, nothing too detailed. We're going to cover, in general, how the nervous system works, the way motor functions work, and how CP interferes with it. Next slide, please. In general, the nervous system is divided into two parts. The central nervous system, which is the CNS, that's the brain. It's your central processing unit. And then there's the peripheral nervous system, the TNS, which is essentially the spinal cord and the neurons associated with the spinal cord. The basic design of the nervous system is you've got sensory neurons for inputs. They relay messages to the brain processes them, and usually that response is, in our case, a motor neuron response. So then an impulse travels down from the motor cortex of the brain, to the motor neuron. So it's a sense, process, response system. That's how it's designed. Let's go to the next slide, please. The major cell types in the brain, there are similar cell types in the spinal cord and in the TNS, but the names are a little bit different. But specifically in the brain, we have neurons, and neurons they're really the heart of the network. The way the neurons are connected to each other is the way the brain works. Supporting that, we've got the oligodendrocytes. If two neurons that aren't next to each other need to communicate, then they have to transmit their messages along the axon, which is that long string you see sticking off of neurons in photos, and I'll show you one of those in a moment. But those require insulation. They make the information, the signal impulse, travel faster down the axon. And the oligodendrocytes produce the material that allows for that. That's the myelin. And then, in addition to neurons and oligodendrocytes, you have the neuroglia, which basically handle everything else. They ensure the blood supply. Astrocytes are a type of neuroglia. They will actually open and close capillaries to allow to control blood flow actually into the brain. They ensure nutritional support of the neurons, and they respond to injury. Let's go to the next slide, please. Here's our neuron. This is actually a lower motor neuron, but the structures are essentially the same. A few things to note. You have the cell body, which is in yellow there, with all of the dendrites sticking off. Those gray bubbles going down the length of the neuron, that's the length of the neuron being the axon. Those bubbles are the myelination. And you'll notice there it says there's a Schwann cell there. That would actually be an oligodendrocyte in an upper motor neuron. Let's go to the next slide, please. And now this is, of course, the brain. I just wanted to point out the quick areas of interest here with CP. CP is a motor disorder. So we're primarily dealing with, if you look at the top center of the photo, you've got the central sulcus there. That's a groove that everybody has that's easily identifiable if you look at the brain. Directly in front of that, we have the primary motor cortex. Now that's where, if you follow the path, that's where the muscles are wired to, directly to. In front of that is the premotor cortex, which is where the higher function processing of motor impulse will go for. There's a certain amount of fine control, spatial organization, essentially determining if you're trying to extend your arm, how far you have to extend your arm. All of that's processed in the premotor cortex. And then down in the bottom right corner of that brain figure, you see the cerebellum. It's not covered, but it's not colored, but that's the cerebellum down there. And like I said, that's some fine-tuning control, motor coordination, that sort of thing. Let's go to the next slide, please. Now, this is a quick cross-section. So, we want to discuss how the brain is wired here. Now, there's two types of brain matter. We talk about white matter and gray matter. Gray matter is what you see around the outside of the brain. When you're looking at the cerebrum, they're dense packets of neurons. And the white matter is mostly the myelinated axons. It's mostly the wiring. So you see those colored lines there on that figure. And I want you to notice that we have the large spaces, the ventricles, in the center of the brain. And look at how that is associated with the wiring. Now, when you've got a case with PVL, paraventricular leukomalacia, you see how having a cyst next to the ventricles can cause trouble with the wiring. Now, with the brain of a developing infant, it'll do its best to wire around it, but you can only do so much as far as recovering from having that sort of a blockage developmentally. Let's go to the next slide, please. There we go. And then this is just showing how a signal gets from the brain down to the muscle. At the top, that's a bit of your brain, beginning with your pyramidal cell, your upper motor neuron. So the impulse travels from the brain through the midbrain to the cerebellum, which does the fine-tuning and control, down into the spinal cord, and then from the spinal cord into the muscle. Next slide, please. Now we're going to talk about the science behind cell-based therapies, types of things we're doing in the lab, and the things we know about embryology, cell development, and the immune system. These are the principles we're working on when we work in the lab. Next slide, please. After you have the implantation of an egg, after fertilization and implantation, cells begin to differentiate. Excuse me. During week 3, you get what we call the primary germ layers. Before this point, before week 3, essentially all of the cells in that ball are the same. The genes, all cells have a complete genome, and almost all of those genes are available. During week 3, you start to see changes, and like I said, you get those 3 primary germ layers. The cells in each of those germ layers started in the same place. They all have copies of all available genes, but different genes are turned on, and that's what happens throughout development. Genes are getting turned on and off by chemical signals, and it's such a complicated process, we're not going to get too much into that, but I just keep that in mind. The process of changing into 3 germ layers and then future development, like I mentioned, signal protein gradients are a lot of it. What you'll get is one cell will have a gene turned on, and it'll start excreting a protein. And then the concentration of that protein goes down as you get further from that original cell. And the other cells respond differently to different concentrations of the protein. That's how you get the head and tail of an embryo. That's how you get a distinction there. The other thing that develops, that assists with development is cell proximity. There are sensors on the cells to detect how many cells are near them and how tightly packed they are. And that process continues into adulthood. When if in the case of cancer, for example, problems with that cell proximity system cause the cells to grow out of control and divide out of control. But it's essentially the same process in an embryo. Cells detect when they're close to each other, and that tells them when to divide or if they should stop growing. Next slide, please. So, back to embryology. From a single fertilized egg, you get cell division, and you get the cells divide very rapidly until you have a solid ball. And the core of that ball eventually opens up, and you get a fluid-filled ball. And that's where we get embryonic stem cells from. I've got a figure in a few slides that will show that. Next slide, please. Just a quick summary of embryology. What we learn is undifferentiated cells can be transferred into different cells, transformed into different cells with particular functions. We all came from the same sperm and egg, and over time, those cells, all of these genes eventually become humans with very, very different cells that do very different things. The control of development is cell proximity and protein signal gradients. And another important thing to realize here is the less differentiation, the less specific a cell is, the more options it has as to what it can become. So if you take an embryonic stem cell, which is from very, very early on in development, if you turn on and off the right genes, it can become any type of cell for anywhere in the body. But it requires more control, and that's much more difficult. With our abilities now, we have to know what every single signal is in development, or we'll get a cell that doesn't work correctly. Next slide, please.
And Cliff, I think it's important for us to say on behalf of Let's Cure CP, you've been talking a little bit about embryonic cells, and currently Let's Cure CP is not supporting any embryonic cell lines. I think Cliff's just been laying the basic foundation for us, and he's giving everybody an outline of how difficult to control the embryonic cells are, and he's just bear with us, but shortly in his presentation he's going to get into the more specifics of the allogenic and autologous cells and how they are completely different than the embryonic cells.
Exactly. Also, in our laboratory, Dr. Carroll's laboratory at Georgia Health Sciences University, we do not work with embryonic stem cells either. Just wanted to make that note. Now let's talk a little bit about immunology. This is the other key with the type of work we're doing. Immunology gives us the opportunity and the greatest obstacle for cell-based therapy. The immune system doesn't just protect against foreign invaders. We're very thankful for that functionality, but it's useful for other things as well. The immune system also detects cell damage. If you get an injury, if you get a cut, the same system that prevents infection also begins to repair that injury, and that's part of the immune system. So it regulates the replacement of normal cells as well. For example, as your blood cells age, they become less efficient and they need to be replaced. And it's the same, and the immune system also regulates how that works. Also, it detects problems with cell replacement. Without an immune system, cancers are very much more common. Patients with immunosuppressive disorders, HIV, for example, are susceptible to many forms of cancer that people with normal immune systems will never, ever see. Next slide, please. One of the most important immune processes is inflammation. What inflammation does is it recruits immune cells, clotting factors, and different repair cells, including stem cells, to repair an injury and prevent infection. And it's good for preventing the spread of a localized infection, but there's heat and there are a whole bunch of very aggressive immune cells present in the inflammatory response, and they can do a lot of damage. The amount of time an area is inflamed, the more damage you're going to get. But this also gives us an opportunity. Inflammation is actively recruiting cells. So if we can use that signal to recruit cells from our cell therapy from a transplant, then we can perhaps get a quicker response, we can reduce inflammation, reduce harmful damage from the natural process, and get our cells in there that will be better than what's naturally available within the body. Next slide, please. The immune response, of course, is also an obstacle when you're dealing with cell transplants. If the immune cells in the body see a transplant as being foreign, it may attack, it may form an immune response. It'll destroy the transplanted cells, and also you'll get a more profound inflammatory response, and that can destroy the surrounding tissue. So we've got to be very, very careful that whatever we do, we don't create a worse immune response than we started with. And so, in essence, the cells we transplant in must either be invisible to the immune system, they must be identified as non-foreign cells, or the immune system has to be suppressed. And of course, we don't want to suppress the immune system unless we absolutely have to, because that will expose the patient to other potential infections. Next slide, please.
And before that last slide that you just presented, I want to make it clear to everybody as well that's listening, with cord blood or the autologous cells that come from your own body, you do not need an immunosuppressant medicine to go along with those, since they are your own cells from your own body. You don't require that. The allogenic companies, I think Cliff will get to a little bit later, address that in the lab, so that any patient can be given these drugs without major immunosuppressant medication, and you don't have to have that issue with the immune response.
Yeah, that's correct. To look just briefly, the immune response, the way it works is essentially your immune system is constantly looking for little bits of protein. All cells have little bits of protein sticking off of them, and your immune system is constantly patrolling and finding strange cells and grinding them up and showing little bits of this to the immune system. And so it basically develops a database as far as what's foreign and what's not. But you can treat a cell so that it won't have any markers on it that will antagonize the immune system. So the immune system will not see it as a foreign cell, or the immune system might not see it at all if you treat it correctly.
And Cliff, there's actually another question on sibling cord blood, whether or not that requires immunosuppression.
I believe in the case of sibling cord blood, it depends. Sibling cord blood, you've got a much higher likelihood of finding a match, and especially among groups with multiple siblings. A single sibling has a good chance of matching, and I've got those numbers. I can look them up.
Was that actually, we were just at a conference about that. It has to do with HLA matching.
Correct.
And you need a certain score out of so many. But there is no, if you do have your sibling's cord blood, right now there are no studies that are using that. However, there are some researchers that are starting to look at that, so that if you could get a score of maybe, say, 4 or 5 out of 6, then you could go forward with the sibling cord blood. But it's just, like, the easiest way for me to explain it would be like if you ever heard of like people getting liver transplants or something like that, and they usually look for their sibling to be the best match. Okay, well that's all well and good, but it's close, but it's not perfect, and the people still have to go on the immunosuppressant drugs, and that's where you end up like losing your hair and things along that line, which you typically see. What we're trying to do here for everybody is put forth medicine that doesn't require any immunosuppressant or look for your siblings' cord blood, because the cells that we can use can either come from the patient's own body or from an adult donor with the cancerous T cells removed.
I just want to add one quick thing. I just heard Joanne Kurtzberg, for those of you that follow along in this industry, know who she is. She is a lead researcher up at Duke University. She actually happened to be in Atlanta last week, heard her speak, spoke to her before her presentation for about 20 minutes as well. And she's the one that has done all the CT autologous cord blood studies right now up at Duke University. Well, according to her, as far as siblings are concerned, they are at least, she said, at minimum 3 years off from looking at that, and also they are looking at some other things in the laboratory. I do have her cell phone number and information. I'm not going to pass it out, but I will say that she or someone from her team will be presenting at one of our webinars in the near future. So stay tuned for that, and we'll let Cliff continue on with this.
Okay. So a brief summary of lessons we learned from immunology. Our research goal is to time the cell therapy when we've got as much natural signaling as we can. If not, then we're going to have to come up with some other type of signaling. We might have to add a natural signal, but add that in with the transplant. There are certain colony stimulating factors that we're researching that we might use, other things like that. But the point is that we need a signal to tell the cells where to go. And we hope that cell therapy will reduce the damage of those natural processes. If we time it right, we get the cell therapy in there in a timely manner, and we start to see repair, then we can reduce inflammatory response and other processes which might cause damage. Next slide, please. Now we're going to get to where everybody's heart is here. The current cell therapy research in cerebral palsy. What type of cell therapy research is being done? We'll go over some of the results and where we're going from here. Next slide, please. Just quickly looking over stem cells. A lot of people throw around a lot of words, and especially when you get into the literature, you start to see all of these potent words. You hear totipotent, pluripotent, multipotent. We're going to very briefly go over what those are. Totipotent means that a cell can become any type of cell, and that's the type of cell you see in a zygote. You have a very basic primitive cell. It's got a complete genome, and it can become any type of cell in the body. Pluripotent cells cannot revert to zygotes. They cannot revert to totipotent cells, but they can also produce just about any type of. They can produce any type of cell in the body, and those are embryonic stem cells. Then from that point, you get into the adult stem cells, the multipotent stem cells. They are slightly differentiated, but they still have dramatic power. We've also seen, for example, with bone marrow cells. Bone marrow cells will normally produce all types of blood cells. From your red blood cells to your leukocytes, your white blood cells. But we have also done experiments using bone marrow-derived cells with stroke and with cerebral palsy, and we have found that the cells can differentiate into neurons. So there's a lot of power in those multipotent cells. Then you also have the progenitor cells, which usually in the organ. They're the cells that can be immediately recruited. If you have damage to your liver, then you might get the progenitor cell in the liver will migrate to that area of injury and become a liver cell. But at that point, it becomes more difficult. You can't take a liver progenitor cell and turn it into a red blood cell. It won't work. And from that point on, you have very limited, because of the genes that have been turned on and off again, you have very limited options as far as what that cell can be without major intervention anyway. And of course, we want to, in developing cell treatments, we want to interfere with the cells as little as possible. We want to make sure that what we have is effective and safe and as natural as we can make it. Let's go to the next slide. Now we're just very briefly going to revisit the cell types. Embryonic stem cells are more proliferative than adult stem cells. They replicate faster, they spread faster, and they can differentiate into of all of the cell types that we can use in the lab. They're capable of differentiating into the most different types of tissue with the least amount of intervention, but they require the most control. Embryonic stem cells come from in vitro fertilized embryos, 4 to 5 days old. Like I said, they're in the blastocyst form. The biggest problem with embryonic stem cells is if they're not properly controlled, if the development and differentiation isn't properly controlled, then you get tumors. And that's certainly not something that we want. Let's go to the next slide, please. The first clinical trial of embryonic stem cells was approved in January of 2009 for spinal cord injuries. Spinal cord is closely related to our work in CP, but there are very, very distinct differences. It's very important to note that something that works in the spinal cord, it's a different environment. It may not work in brain, and it might not work. It may work in an adult, but it may not work in a child with CP. So it's very, very important to watch these other areas of research, but realize that there are big differences. Another note about the embryonic stem cells is we don't have a proven usage up to this date. We've tried a lot of things, and there have been some reports of positive results in uncontrolled overseas experiments, but no proven usage that is scientifically recognized in the United States. Next slide, please. this is just a quick slide demonstrating embryonic stem cells becoming any type of cell. So we have the blastocyst, and the cells from the blastocyst are taken out and put into culture. And under certain conditions that we manipulate in the lab, we have demonstrated they can become neural cardiac cells, cardiac muscle cells, blood cells, many different types of cells. Next slide, please. Now, adult stem cells, these are what we primarily work with in our lab at the Medical College of Georgia. They're undifferentiated cells in a specific tissue, like bone marrow cells. We work extensively with bone marrow cells. They differentiate along defined pathways, which means we know a lot more about how they develop, how they turn into different cells, all of the different signaling in that process. And manipulation might allow differentiation into other lines. For example, we've demonstrated, with our colleagues, we've demonstrated that we can get bone marrow-derived cells to turn into neurons. Next slide, please. Here's just a quick slide showing some of the different pathways with the bone marrow cells into liver cells, skeletal muscle, marrow stromal cells, which are a particular subculture of bone marrow cells, have been shown to form epithelial cells, which are skin cells, cells that line the gut, that sort of thing. Neurons, cardiac muscles. They'll also form fat cells. Let's go to the next slide, please. Most commercial development right now is in the area of adult stem cells, and some companies are initiating clinical trials at preclinical trial. The clinical trial stage is quite complicated. There are a lot of different steps to it, and we'll cover that in a bit more detail a bit later. Next slide, please. A few of the conditions that can be treated currently with adult stem cells: leukemia, lymphoma, hemoglobinopathies, which are abnormal hemoglobin formations, bone marrow failure syndromes. We replace bone marrow with a transplant, certain immunodeficiency syndromes, some metabolic disorders, and of course there are some neurological disorders that are being treated with adult stem cells. Next slide, please. MAPCs are another type of stem cell we're looking at. These are multipotent progenitor cells, adult stem cells. Verfai in 2003 demonstrated that bone marrow-derived stem cells can become many, many different types of cells. And since then, the company Athosys Incorporated, who we're working with, has been working with us and a number of other labs in using particular types of MAPCs in the treatment of cerebral palsy, spinal cord disorder, and stroke. Next slide, please. Induced pluripotent stem cells are fairly new. We refer to them as IPS cells. They are stem cells that we have that are modified slightly in the lab. They're exposed to certain chemical signals to put them into a more primitive state. So they behave much more like embryonic stem cells. They express many of the same genes and proteins, and the list is there. That's more for the scientists, not too much to worry about there. But the capacity and how much control is going to be necessary is still being assessed. Like I said, they're very, very new. We've started to work with some of these in our labs, and we've got a colleague who's very interested in these cells. But we've only just begun working with them. Next slide, please.
Cliff, what are these cells derived from, and what makes them like embryonic stem cells?
They're like embryonic stem cells because they are modified in the lab. There might be more iPS cells in the future, but these are, I believe the ones we're working with now are bone marrow derived. Let's skip to the next slide, please. We'll come back to this one. Now we've talked about cord blood, and cord blood is a very, very hot topic. Cord blood, they're very highly proliferative cells. They're non-controversial, unlike embryonic stem cells. Decreased rates of graft versus host disease, that indicates that there's less immune rejection. And then the HLA mismatches are, again, part of the immune system. HLA, we've discussed this before, but HLA is essentially the typing, the immune typing they do for all transplants. And they're more tolerant of mismatches than most whole organs, heart, lungs, liver, etc. Let's go to the next slide, please.
And just to be more specific, if you're receiving your own cord blood, there are no mismatches, and this kind of goes back to the original question where there is the potential to go up to like a 4 out of 6 match or a 5 out of 6 match with a sibling, or there's actually banks that can do HLA matching that might even give you a better match than your own sibling cord blood if that's what someone is truly interested in.
Correct. And on this slide that we just popped up, it's a 25% chance of having an identical match with any one sibling. So we can go to the next slide. Now this is actually from our own laboratory. We got some very interesting information early on. We were using a rodent model. Our rodent model for CP is an animal that has a hypoxic ischemic injury at 7 days. What we do is we constrict blood flow, or we ligate, cut, or tie off one of the carotid arteries, expose that animal to 8% oxygen for a limited amount of time, and that will produce an injury, very similar to a birth asphyxia injury. What our very early study was to just look and see what the brain did by itself. Were there new cells in brain? Up through the 1990s, it was believed that the brain did not recover. Once the fetal development was over, that the brain would grow and expand, but dead cells, there was no response beyond inflammation when cells were injured and died. With this, we injured the animals and then looked at the brains at certain different time points. And we noticed that, yes, there were new cells. BRDU incorporates into the DNA of cells. And so if we injure the animals, then inject them with BRDU, only new cells will show up. So you look at the left side versus the right image, left image versus the right image there. You have the undamaged side, and you have the damaged side, the injury side, and there are a lot more cells on that injured side. So there's definitely a cell response. There's definitely new cells on the injured side of the brain. Next slide, please.
Cliff, can you explain that a little bit better? So, what are the little bright red dots on the left side versus the right side?
Sure. This is what we call immunohistochemistry. We get fluorescence. Whenever we look for our protein target, we get this fluorescence, and these bright red dots are that fluorescence. So the dots that are lighting up are brand new cells. The DNA has incorporated this protein, and we've attached a fluorescent marker to it, and so they fluoresce like this. So on the left side, you've got very, very few new cells, and this is a brain that has not been damaged. And in the right picture, the cells, there are many, many, many more new cells in the brain that has been damaged. So there is a response. There are cells recruited to the site of injury in brain damage. And then here we have the rodent brain is a little bit different from the human brain, but what we're looking at here is the hippocampus in a rat brain. That's these two large lobe structures in the center of that top photo. And what we wanted to show is that some of these cells, these new cells that are recruiting, are in fact neurons. So again, this is immunohistochemistry. We looked for the BRDU, which would indicate the new cells. That's the red. In frame C, that's one cell that's labeled with the BRDU. We also looked for a protein called NUN, which is a neuron nuclear antigen. And what that indicates for us is a neuron. And if you'll notice in frame D, that same cell is also lit up in green. These are under different color filters. Frame E is a counterstain, which shows us that it's a real cell. It's not just a splotch of color that shouldn't be there. So frame B is actually the composite of C and D, using a triple filter, which lights up both at the same time. So what this tells us is that cell in frame B is both a new cell because it's BRDU positive, it's got red, and it's a neuron because it's stained green in D. So you see that composite in B. That's both a new cell since injury, and it is a neuron. Let's go to the next slide, please.
I think just so everybody understands that, this is huge because what you just showed in that last slide is through regenerative medicine, you actually can create new neurons in the brain.
Yes. It happens, it's actually a natural process. In this particular slide, we did not intervene. So new neurons do form in the brain after injury, without question. So we need to enhance that process. So let's go to the next slide. This is what we call confocal microscopy. What this allows us to do is generate a 3D image, a three-dimensional image. Normally looking under a microscope, it's really two-dimensional. At any one time, you're only looking at one plane. But the confocal allows us a 3D image, again, to identify that what we're looking at is really a cell and not a splotch or something artificial that happened during processing. So what these are basically the same 3D reconstructions. If you look at inside the purple box in the margin on slide A, that's actually the three-dimensional depth perception for that marked cell. And that's a BRDU and NUN positive, so that's again a new neuron since injury in the granular layer of the hippocampus. The granular layer of the hippocampus contains a lot of neurons. Now let's go to the next slide, please.
Did you want to go back to the rat?
It's all right. I think I discussed it, so I think we've covered that. That's fine. Looking at MAPC transplantation and neonatal hypoxic ischemic injury, we've seen with different behavioral testing, we primarily in our lab work with the rotorod. The rotorod is essentially it's like a treadmill. The rats are placed on a round cylinder, and it accelerates. It's controlled by a computer, and it accelerates, and we measure the amount of time that the animals stay on as it accelerates. And we also look for problems, weaknesses on one side or the other, perception problems, whether or not they can detect where the walls are, because they'll actually move back and forth on the rod as they're walking forward, so they can judge their hand and foot placement. So we look for different problems there with sensory and motor output. But treatment with the MAPCs, we've seen improvement between untreated animals that have been injured and animals that have been treated with MAPCs. They stay on the rod longer. They have better spatial perception. They're better at placing their feet where they need to be in order to stay up, and also they're more aware of their body position, which is excellent. We found that a very small number of MAPC cells become neurons naturally, and we're working on improving those numbers. We've also noticed that transplants seem to enhance the survival of neurons that are already in the tissue. So not only do our transplants become neurons, but when we do a transplant, it improves the outcome for the cells that have been damaged. We have more of them surviving. The MAPCs are well tolerated immunologically. We've actually been able to transplant between species, between mice and rats, for example, with no immune response, which is excellent. Also, MAPCs work well when they're injected intravenously. We don't want to do neurosurgery on a child if we can avoid it, and so we're looking for a cell therapy that we can inject into the bloodstream, put into a drip, as opposed to something we would have to physically inject into the brain. So it's very good that they work just as well IV in our animal models. It's a good sign. The only downside is that because of signaling as it is now, because of what we understand of signaling in CP, we might need to do those transplants very soon after the injury. Next slide, please. Now let's talk a little bit about what is necessary to get the research out of the lab and into the clinic. Let's go to the next slide. The FDA, the Public Health Safety Act, Section 351, regulates the licensing of different therapies, drug applications, etc. And so our cell therapies are subject to that regulation. Human cell tissues, cell products, they're generally treated the same way, and they have to go through the same process as a synthetic drug. Next slide, please. The major checkpoints, what's the FDA looking for? Well, they want to be sure that any cell therapy we develop is safe, that it's pure, and that it's potent. We want to make sure that it works against the disease we're targeting. We want to make sure that it doesn't create negative effects. We want to make sure that there isn't any junk in there that isn't needed. In our specific case, we have to be sure that we have adequate screening against infectious disease from a donor. In many cases, we're going to be getting these cell treatments are going to be coming from siblings. They could be coming from other relatives. They could be coming from blood banks. So we have to be sure that in the creation of our cell therapy that we screen against infectious disease from any donors. We want to make sure that the process of developing, going from our raw materials and developing that end product prevents contamination. We don't want any junk in there that could be a problem. We also want to make sure that we don't, in the process of handling the cells, damage those cells. So the FDA wants to, looks over our shoulder to make sure that we don't develop a process that could introduce that type of a factor. We also want to disable any mechanism which would allow an undesirable effect. This specifically has to do with the cell signaling, especially for the differentiation. If we're going to be manipulating cells, we want to be sure that we treat them in a way that they can turn into something else. We want to make sure that we understand how the cells are going to develop and what they're going to be, and that there's no chance for them to become anything else. And like I mentioned before, we want to make sure that they're potent, that they actually affect the targeted condition. Next slide, please. That about covers it as far as our presentation goes. But I just wanted to give some acknowledgments. Dr. Hill works extensively with us at Georgia Health Sciences University. That's really our sister lab. He does research into a lot of things, but he's primarily a stroke researcher. Dr. Hess, who is actually our chair. Dr. Carroll, who's my immediate supervisor. A lot of the research by our lab that's been presented here It's been supported by the National Institutes of Health, the American Heart Association. Athos has incorporated, and of course, Let's Cure CP. There's a reference there for further FDA regulatory information. It's also available from other sources as well. And also a credit for the slides in my Neuroanatomy section are from my favorite anatomy and physiology textbook, and that's the credit right there.
Well, thank you very much, Cliff. And I think at this point, if there's any callers that have any questions, if they can go to the top of their viewing bar where it's green and just put your icon on there, and you can click on that maybe chat button and say you have a question, and then Kevin, you might be able to bring them in, right?
Yep. I think there's also a way to raise your hand. I can do either. All right, Rachel, hold on for just a second. We'll bring you on the line. Go ahead, Rachel.
Can you hear me?
Yep. Yep.
Mm-hmm.
Okay. Okay, I hope this isn't too straightforward. Actually, first I want to say thank you so much. This has been absolutely wonderful. But as of right now, what we are thinking about doing Dr. Bumgar's study for our 2-year-old who had a stroke during labor. And basically what I want to know is if we were to try to do Dr. Carroll's study, would I be able to use my bone?
Hello?
Rachel, you cut out.
I think what Rachel is trying to ask is whether or not she would be able to use her bone marrow, but of course my question is, why would she be able to if the child
and I think we know the answer to that. With Dr. Carroll's study with athersis, they can't tag a particular person's bone marrow, correct, Cliff?
Correct. Right. At this point, we're not doing that.
Right. And then with Dr. Baumgartner's study, the one that he's proposing, that would just be taking the bone marrow from the child's own hip bone and then just readministering that, which is the cleanest, safest, most pure way to do it. The key to what we're dealing with cerebral palsy is that every child's going to be different, and I think the screening for the child's going to be very important to make sure that there aren't any underlying conditions with the child, because if you do use your own bone marrow and you have a degenerative disorder that hasn't been brought forth, your own bone marrow will not do any good, and that's when it's so important to have the allogenic cells available for those patients as well.
Okay. We've got another question from Lisa. Lisa, go ahead.
13. Hello?
Go ahead, Lisa.
Yeah, I have a daughter who's 13 who has mild to moderate CP, who's pretty high functioning. I was wondering if there's anything that they're working on for children like that.
Well, that's what we're doing at Let's Cure CP. All of our children are older as well.
Okay.
And the nice thing that I've heard from many physicians is that there's no limit to neuroregeneration, which means that I thought that the clock was going to be ticking on my son. He just turned 7.
Okay.
And that I needed to do something because there's going to be like a timeline where you're not going to be able to do anything.
Right.
And, we found these physicians that are willing to do the studies on what your child has and my child is chronic CT. And so these studies that we are funding will be for chronic cerebral palsy patients.
Now, Ed, I'd like Cliff to just address something that was in one of the slides. He mentioned that, here, I'm going to see if I can go back to it. Transplants may need to be done sooner after an injury with the MAPC. Cliff, can you kind of address that in relation to what I was just talking about?
Sure. The issue there has to do with signaling. And The model as we currently have it, the rodent model, which is where we have to start, the rodent model isn't really good chronically. The biggest problem we have is that most of the signaling that we intend to use is gone in a very extended chronic model. If we begin treatment early, it extends that window, but that window, that signaling disappears over time. We are also looking into improving our chronic model so we can take a closer look at the signaling and create the necessary signals later. But I think it's going to be part of the normal development of things that unless we have breakthrough out of left field, which often happens in science, but unless we have a breakthrough out of left field, I think we're going to be able to treat newer cases sooner than chronic cases. But as I've said, the potential is most certainly there. It's just a question of really nailing down the signal.
That was published last week where they're using lectin, which is a product produced by a plant, and they're putting it at the injury site, and the cells will adhere to the lectin and stay there. So that's going to be another interesting breakthrough. There's so much to be done and studied with this particular topic, and the potential is so incredible that that's why we're doing what we wanted. What we're doing with everybody here to promote these studies and move the field forward.
I want to jump in here for 1 second because I don't want to confuse the issue with the question that was raised about the 13-year-old, and I see that we have another question about the same issue about funding the chronic model, and I think I speak for all of us when I say we all have older children. My child is 9. What Cliff was referring to is, I will say that all doctors across the board that are doing this research, everyone understands that when you treat a child as soon or as close to injury, generally speaking, you see a greater result. So we get that. However, what QCP is looking at funding, and we want to make sure that you guys understand, and we are adamant on that, at least for this initial round of funding. What we're looking at is Dr. Carroll has a chronic mouse model, and he also has an acute, and we are looking at funding more that chronic model, just so you guys understand that, which is the patient that has, had CP for some time. I'm not promising it's 15 years, but, at least somewhere in that, maybe 18 months is the low as the FDA will let us go to, is up to, maybe 10 or 11. And again, I don't want to say we won't even, he won't even accept a child at 13 or 14. It's just, honestly, as far as where the parameters are placed, really is up to the researcher. And then, of course, it's up to us whether or not we want to put money behind that project. But, we have to understand that these are their projects, and then we choose whether or not we want to fund it. But with Dr. Carroll and also in speaking with Dr. Baumgardner, they have assured us that, our chronic children will not be left out.
And was that as you were addressing, children and the studies, there's a question about whether or not studies are currently enrolling patients.
Right. We can address that right now and let you know that, unfortunately, the answer is no. And we'll talk to 2 different studies, but for the purposes, let's address what's going on at Medical College of Georgia right now. This study is what's called preclinical, and Cliff can give you more background on that. But basically what preclinical means is that it's being studied in laboratory animals. What we have committed to Dr. Carroll and his team is we want to get them $100,000 because that will fund preclinical and then for these rats in the chronic model, and then you present that data to the FDA, you file for approval on an actual human study. The nice thing about the Athersys cells, which is what Dr. Carroll and his team is working on, is they have been used in over 10,000 patients, and maybe you can talk about this, but I don't think anyone has died from the atherocyst cells. Am I correct?
No. No.
So, you know, that...
More importantly, I don't even think there's been any tumors that have...
Right.
The atherocyst cells are the bone marrow-derived adult donors, and they process them in the lab, and then they match them up, and they're taking the cell and treating it so that it's safe for any individual. So it's like a type O blood, where no body would reject it, and there's been no deaths, and I don't even think there's any hint of any tumors forming. And like Lizette said, it's been in over 10,000 patients so far, and for our trial to get up and running, they actually have to. The Atherocyst already has a trial going treating adult stroke patients, and cerebral palsy would fall into the same genre as the stroke patients. And so after the adults are treated with that, then they'll start the trial for cerebral palsy. And Dr. Carroll already has his protocol ready to go for acute cerebral palsy treatment. And what we're funding would be the chronic stage of cerebral palsy. So what Dr. Carroll's study is going to be for the chronic older patients that have had the injury for a longer period of time. And once he can hopefully show benefit, then we can get that treated in a phase 1 clinical trial. And then what Dr. Baumgartner is attempting to do, he has already done the trials on traumatic brain injury patients, and he's going to try to submit to get a chronic cerebral palsy study going using autologous bone marrow cells, which means coming from your own hip, without having to do the preclinical work based on his previous studies.
Right. So the safety has already been proven. So, and as far as timeline for that, neither are currently enrolling. Dr. Baumgartner needs to submit his IND, which is investigational new drug, which is basically, even though this isn't a drug, it falls under that category, and that needs that to file that submission generally takes a good 6 months, at least, maybe a little bit longer, and then he could start enrolling in the study. But the bigger issue is this, is that that study is going to cost about $35,000 per child, and of course the cost will not be passed on to the parents. That's what we are trying to raise money as well for that particular study, because we're talking a minimum of $500,000, and we have already been down the road of discussing this with the NIH, which is the National Institute of Health, if I'm speaking in acronyms for people. And what the NIH has told us, which is across the board, not just this CP category, is initial studies are generally privately funded. Once phase 1 is complete and they say, hey, there might be something here, that's generally when these researchers submit for grants and they're able to get dollars for their phase 2 studies, which tend to be in the multiple millions of dollars. So that's kind of where we're at.
Okay. I don't see any additional questions.
Can we go back to Cliff's one anatomy slide, and I just want to say a few things about that one, about the neuron and the myelin sheath.
Mm-hmm.
Right now, the doctors, when they're injecting these cells, they're typically doing it through an IV. There are some studies that can interject the cells. Keep going back. Okay, perfect. There are certain trials that can actually inject it into the spinal cord. And that gray little strand of, looks like little, bubbles, I guess, that's your myelin sheath, and that's what's carrying the information from the neuron to the other neuron, and that's what they're calling the white matter. And what the scientists actually think, and they're not certain on this, I mean, even though they're able to grow additional neurons, what they think what really might be happening is that they're just the myelin sheath is being repaired as well, and that's more difficult to prove under the imaging, but they think that they're improving that myelin sheath, so the information that's in the one neuron that goes to the next one is actually transferred more efficiently. And the way I equate it would be like if you had to have a garden hose with holes in it, and as the water goes through, and there's some water that's shooting out, so it's not an efficient flow of through the hose. Some of the scientists think that they're either repairing the myelin sheath or the peripheral nervous system, as well as the potential to grow additional neurons. But all this still needs to be studied in more detail.
I think we have another question from Jen Ju. Are you there?
I'm here.
Go ahead.
Thank you. I joined a little bit late, and I just wanted to just get an idea on these studies, kind of get an idea of which may be most appropriate for my child versus other children and such. I know there's a couple different ones out there with a little bit of a different focus. So, for example, for my kiddo who was a twin preemie, has been diagnosed with spastic diplegic CP, uses a walker and things like that. She's actually gone through a number of rounds of Botox injections and recently had surgery for muscle lengthening. But I wanted to see for where she is, because I know how she's treated is different from the treatment of a stroke patient, and which of these trials we feel that she would best fall under. Does that make sense?
Yes.
Okay. It's been a long day. My transmission went in my Pathfinder today, so it's been a long day for me. So hopefully I'm still making sense.
I'll address that for a second here. Hi, Jen. It's Lizette Dunay.
Hi, how are you?
Hi, good. I think, that's a little bit of a fine line, and we need to be careful, those of us here on the phone trying to play scientists, because
sure.
as we said, our job as an organization is to find the studies that we think will help the most people,
right,
and then help fund those studies. But
we can't predict.
Hate to tell people, yes, your child will fit in this category, and then your child will fit in that category, and and then have very disappointed and upset parents if that's not the case. But
sure.
Just to kind of tell you where these scientists are, kind of what they're telling us, and maybe Cliff might have more information on where they're thinking with a rat model, but it seems like everyone likes the stroke babies, the babies that have had brain bleeds. And the reason it seems that most researchers like to start off with that patient population is because it's very easily identifiable. As we all know, about 50% or more of CP patients, they have very clean MRIs, and they're not really sure why they have CP, but they do, and they present with CP. So we really need to be careful with that. And Cliff, do you have anything to add to that?
The rodent model is a little bit different. At this point, at least in our lab, we're more focused on the neuron death, which may or may not be associated with hemorrhagic transformation with the brain bleeds. But the model is just a little bit different.
Okay.
So the clinical, as the clinical studies are designed, when they get to the clinical phase, those things are taken into account. Dr. Carroll and I have discussed it from time to time. But the model as it is in the lab with the rodents does not include those hemorrhagic transformations.
Okay.
And quite frankly, I think it's we have to. we're not going to really know a true answer until the studies are done anyway. And, right now we need to, get this to the point where we can start treating humans, follow them, and see what data comes back. But the only thing that I have to go back on is that, every child with the cerebral palsy, I think it's very important to have that preclinical workup before entering the trial to make sure that there are no degenerative disorders along with the cerebral palsy, because if you do use your own bone marrow and you have a degenerative disorder, then it won't work.
Right.
So, that's why it's important to have these different cell lines. And, we do need to, fund these trials, move them forward so that we can learn from them. And so, 3, 4, 5 years from now, I think we can answer your question more definitively.
Sure.
And saying, based on the work of Dr. Carroll or Dr. Baumgartner or Dr. Kurtzberg or one of the leading experts, we could say that we do have the data, but quite frankly, the data, we have to make the data. And, that's why we're doing what we're doing, because right now we really can't answer that in a straightforward yes-no answer.
But they basically all involve use of a child's own bone marrow.
The autologous cell, yes.
Okay.
The allogenic cells, they are healthy adult donors' bone marrow
Okay.
that is given to companies like Athersys, and then Athersys will process the cells so that they're acceptable to every human, and that they won't be rejected. A lot of this presentation in the beginning, which will be put back on the web, on our website, kind of goes through a lot of the things that can go wrong. And, they're talking about immunosuppressant drugs. And, what we're trying to do is fund safe, clean studies that don't require the immunosuppressant medication, and the body will not reject. And the Athersys cells have already been processed so that the human body will not reject it.
Okay. And so the focus would be on getting that money raised to be able to start
Right.
funding these trials, which I completely understand. I worked in pharmaceuticals part-time and understand how everything works with the FDA and such, and how many battles and obstacles that you can face. So, okay, so that's why it's important for us to kind of step in here as parents and do what we can.
Right, and we've identified, several of the doctors that can do the work, that are the leaders in the field, and like you said, with the support of the community and the people that understand and want to support, this type of research, we will get it done.
So, there's another question kind of about the efficacy of the different stem cell therapies available, and I think you kind of spoke to it in some of your discussion with Jen there, just that we've got to get the studies done to figure out what the efficacy is. Yeah, we at this point we don't know. I mean, to be point blank with you, for someone to sit there and say, this is what's going to happen. The trials haven't been run in the United States. I mean, all we have to go from is like what we've seen on the internet, and as a cerebral palsy parent, you want to hope and believe the best and all that great stuff. But when it boils down to it, what Cliff was saying, in our medical profession, they want to have some proven, legitimate studies to bank on, to give claims, and there have been institutions that have treated 100 to 200 patients, and they never tabulated any of the data, and why did they do that,
Well, there is a double-blind study, guys. What that means is one arm of the study, the patients are getting the real thing. The other arm of the study, the patients are getting placebo. And that's going on right now in Joanne Kurtzberg's lab, Dr. Kurtzberg's lab at Duke, with the autologous one's own cord blood. And first half of basically it's the first infusion You either get the real deal or you get the fake stuff, and then you come back in 6 months. If you got the fake stuff, you get the real cord blood, and vice versa. So that is double-blind, randomized study, and that's basically what the medical community wants to see. She is about 9 months into that study. I know that that Dr. Carroll is also right now working with autologous cord blood as well. And I know Dr. Cox is doing the same at the University of Texas, and Dr. Carroll, as you all know, is at Medical College of Georgia. So that the studies are happening, and it will not be just anecdotal, parents going on national TV saying, it's a miracle, my child is running a marathon. So this will be actually documented medical research, and I think that's going to benefit the whole CP community when those results are. So probably sometime mid-2012 is when we will have some hard data.
I think we've got a couple more questions, and I think we're going to have to wrap it up because we are getting close. I'm going to go ahead. Fran, I see that you've got a question. Why don't you go ahead? Fran, you should be off mute. Oh. Sorry, Fran, I'm sorry. I hadn't had you off mute. Go ahead. Try again.
Okay. Well, I've been reading a lot about stem cell research, and tonight is the first night that I heard about the embryo stem cell research and about tumors. But EJ, my grandson, is 3 years old. He is starting to walk, so we really are very, very beneficial compared to some of the rest of them. Without braces and without a walker. He can't speak, he can't chew or swallow anything. But they've now just declared him deaf, and I've been reading on the cochlear implant because it was a little disappointed because they said he had to be 100% deaf before they could receive that, and he's like 100% in one ear and 40 in the other. And so I've been pushing for that, but they're not letting that happen here.
I can't talk about the cochlear implants because I'm not as familiar with that, but I can tell you that there is some preliminary research showing that stem cells do go to the part of the brain and actually repair and regenerate hearing loss.
And I believe that, Lizette, because really he's not, when I go to the doctors, we are truly, truly lucky. I mean, he's walking. Of course, he looks like a little drunk man, but he is walking without his braces and his walker. And so he's a typical little boy getting into everything. And we're just real happy with his progress. But one thing I wanted to add for people to know about what we went through, and maybe somebody can learn, when EJ was 2.5 years old, this child threw up from morning to night, and he would gag himself. And every therapist, every doctor told us that is what cerebral palsy victims do to self-stimulate themselves. And that he was making himself throw up so that he can enjoy himself and play in his vomit. Come to find out, we got a pediatric endocrinologist who said that was all such a BS, that the baby for 2.5 years was allergic to milk.
Oh, I'm sorry to hear that.
So, I mean, that's just something that, if you hear of any case or anybody else, I mean, the kid had 5 therapists and 7 doctors, from a neurologist and it finally took a pediatric endocrinologist, Dr. Terry DeClue, who got all those reports to put it together, and it took us like 6 weeks to keep him lactose intolerant, and now he's growing taller, so he's still thin, but he's eating. So that just may be something to throw in for parents that, may go through the same thing. But I'm hoping with the stem cell research and maybe that cochlear implant, he'll be on his way because he's a very bright child. And when I look at the others, we truly are blessed.
And if I'm not mistaken, I think Dr. Baumgartner is doing a autologous stem cell trial for cortical deafness, right?
Yep, he is. He's very interested.
Beautiful. Yep. Well, he has his cord blood and all that. And I want to thank all of y'all immensely for everything that you've done. And I'll go let somebody else take some time. Thank you.
You're welcome. Thank you.
Let's see. The last question that I see up here is Athersys helping with funding for Dr. Carroll's study, and what phase is his current study?
Yes. Athersys is definitely contributing. They also are on an NIH grant with us to secure more funding. We're with the Athersys itself. I can't go into too much detail, but we're going through the preclinical phase. We have already done the safety study in the rodents, which is a large set of animals that are treated with the cells, and then a bunch of FDA's pathologists come in, or a third-party pathology lab come in, and they look at all the animal tissues, look for anything abnormal in those tissues, and send their data directly to the FDA. And we've already been through that. We're doing some other fine-tuning studies with Athersys, but they are definitely supporting us financially and also cooperating with us in getting research funds wherever they can.
So just so everybody understands, I think what you're saying is your chronic CP rodent model has already been treated with Athersys cells, and the third party has come in and analyzed your animal model, and so they will be getting back to you to say whether they see improvement or not, and then once you hear back from them, that's when you can apply for a phase 1 clinical trial, right?
Well, the chronic model, we are doing the safety study. With the acute model, we've completed the safety study, and we have had no negative results. Right.
Exactly. So that one we're fine on. But okay, so exactly with your chronic model, have you started to treat the rodents right now with the Athersys cells in your chronic model?
Yes, and we're collecting that data.
Okay, so you're collecting the data now, and then you have to have the third party come in and analyze the animals, and then you would be able to petition for a phase 1 clinical trial.
Exactly.
Right. So for a time frame for our audience, that'd probably be about 6 months, maybe?
I'm trying to remember exactly what the time frame was for the acute. That sounds about right. That sounds about right.
Okay, Ed, why don't you go ahead and just take us out?
Well, I want to, first of all, thank Cliff for his wonderful presentation, and thank everybody for their wonderful support. Without the support of the people out there, we wouldn't be where we are today. I hope everybody found this very educational. I know I learned a lot about things as well. And what we want to do with this first webinar is just get everybody a strong, foundation in what regenerative medicine is and how we can move this forward for cerebral palsy. So Cliff was gracious enough to put this wonderful presentation together so that we all can learn from it. And then what we're going to do is we're going to put this presentation on our website so that people can access it at any time. So if you want to tell friends, family, therapists about this meeting, hopefully it won't be more than a week or two before we can have it on our website, and you can direct people to, so they can educate themselves about it. And then what we want to do in the following months is have Dr. Carroll on, Dr. Baumgartner on, somebody from Duke University as well. So now that you have the strong foundation in the basics of regenerative medicine, we can go into even more detail with Dr. Carroll and Baumgartner and the folks from Duke in the upcoming webinars. And if Lizette wants to add anything at this point, all I have to say is thank you very much for your support, and that's it from my side.
That's it. Thank you again to Clifton Medical College of Georgia or Georgia
Health Sciences University.
I'm going to have to get used to that one. And also, I want to say thank you to Kevin Metzger, who is one of our board members who helped put this together. Thank you very much.
Thanks. And I guess I'll just throw in that we can always use your support. It's through your help and through funding that we're able to do these studies and put these things together. So even if you can't give directly, if you want to do something, recently Fran and her daughter put on an event that raised $9,000 for the organization. So I want to say thank you to Fran for that. Just those are the types of ideas. I mean, it was a roller skating event that they raised $9,000 for. So those are the type of things that are just tremendously beneficial to us as an organization. So thank you, Cliff, and Adam Lizette for all your knowledge in putting this organization together from my perspective, too.
All right. Good night, everyone.
Good night.
Good night. Thanks for having me.
Bye-bye.